So you just watched a video that talked a little bit about linked genes and why we see certain characteristics that can get passed down together more than others. Now, that distance between those genes, as you know, the further apart that two genes are, the more likely it is that a crossing over might occur. Now, even if genes are fairly close to each other, there's always the probability, although it might be very small, that a crossing over event might occur there. So let's take a look to see how we might determine if genes are linked. So purebred lines of tortoises, domed shell, checkered shell pattern, are mated with pure tortoises with tall shells that have a swirl shell pattern. So a domed shell, big D, is dominant over the tall shells, little d. And the checkered shell pattern, big C, is dominant over the swirled shell pattern, little c. So the resulting F1 offspring, that's the first generation after this mating event of the purebred lines, are all heterozygote for both traits. A test cross was then done where the female F1 tortoises were crossed with the male tortoises recessive for both traits. So let's take a look at these questions. What are all of the expected phenotypes, the probabilities of each phenotype, and the actual numbers of offspring expected for each phenotype of the F2 generation resulting from the test cross, based on a total of 1,000 F2 offspring? So let's take a look here. Let's do a little recap of what we're dealing with. So we have a parental generation that we know what the genotypes are because they were purebred for particular traits. So we know that the parents were a cross of big D, big C, times little d, little c, okay? That means that the F1 generation was all big D, big C, little c. And so they did a test cross, which means that they crossed this genotype here with a completely homozygous recessive test cross individuals there, okay? And so now what we need to figure out is what are the proportions of the individuals from this test cross. So again, we can make our little Punnett squares. You should be getting very good at these, where we look at our D's and we can come up with our proportions of what we're going to see. All right, so here we have the domed and checkered, and we can come up with our checkered situation here. Okay, and we can come up with all of our proportions and multiply them out, and what we'll find is that we are going to get domed and those checkered individuals at a rate of 1/4, okay, or 25%. And that should correspond out of a total of 1,000 individuals to 250. Next, we're going to have tall and swirled. And if we count all of our proportions, just like we have been doing for these Punnett squares, I'm not walking you completely through the entire Punnett square because we've done several of those by now. What we're going to see is that, again, it's going to be a total of 1/4 or 25%, which out of a total of 1,000 individuals should come out to be 250. Then we have our tall and checked, which again, if we do our probabilities and multiply those across, is going to be 1/4 or 25%. And so we would expect to get about 250 individuals if we had a total of 1,000 offspring. And then finally we have domed and swirled, which again is going to equal 1/4 or 25% or 250 individuals. Okay? Now, let's keep reading. So here are the actual results. So you or a scientist actually sets out to do that test cross. And then they count out exactly what the offspring look like, how many of the offspring fit into each of the different categories. And so here are the numbers you got. 444 of the offspring were checked and domed. 427 were tall and swirled, so on and so forth. So what are the actual percentages of the above phenotypes? So we're going to divide 444 over 1,000. And so we're going to get just about 44% for this first trait. I guess I'll only write it right here. We're going to have about 44%. Okay. For the tall and swirled, we're going to have about 43%. If we divide 427 over 1,000. The tall and checked is going to be just about 8% or 7.5, so somewhere around there. And the domed and swirled are going to be just about 5% or so. Okay, give and take a couple of tenths of a percent or whatnot. So now the question is asking us, are these two traits linked or unlinked? And what we need to do here is compare what we were expecting to get with what we actually got. And if we actually got what we expected to get, then we know that the traits are not linked, okay? If it deviates, if our observations actually deviate and are different from what we expected, it is very likely that these traits are going to be linked. And when we compare that we should have gotten 250 individuals of each of these different combinations, and we compare to the numbers that we actually got, we can say, oof, absolutely, these are going to be linked. There's something going on there, okay? Now let's keep going here. Which of the above phenotypes are the parental phenotypes, and which are the recombinant phenotypes? Now the recombinant phenotypes are going to be those phenotypes that the crossing over event occurred, and they created these new pairing of alleles that aren't very common or didn't exist before, okay? Or weren't potential allele combinations that existed before. And the easiest way to go about this is that the parental phenotype is always going to be the phenotypes of those, let's go back up here, of those numbers that were the highest. So in this case, the domed and checked and the tall and swirled. And that's because not a lot of crossing over event is going to be occurring between these linked genes. And so the majority of them are going to keep the same combinations that the parents have. Okay, so that's an easy way to figure out what your parental genotypes are and what are your recombinants. So your parentals are going to be the domed and checked, and and the tall and swirled. Now your recombinants, I'll just write them down here. Your recombinants are going to be the tall and checked and the domed and swirled. Again, the kind of two combinations that are the least common or not seen as frequently. All right, finally, let's go to this issue of map units. So a map unit is basically something equivalent to like a kilometer or a meter or a yard. It's a measure of distance between two genes, okay? And the question here is wanting you to figure out how many map units apart are the two genes on the chromosome, and to draw out the chromosome and label the two genes and their map units apart from each other. So how do we do this? Well, the first thing to keep in mind is that when we look at the map units, what we are basically the way to figure out what a map unit is, so a map unit is going to equal the number of recombinants divided by the total number. So if we go back to our numbers here, we know which ones are our recombinants, 75 plus 54. So we're going to have 75 plus 54, which is 129, divided by the total number, which is 1,000. And what we're going to end up, you can either multiply these to give this as a percentage or keep them as the map units. We're going to basically end up with 13%, right? If we're multiplying it, it's going to depend, or basically 13 map units. All right, and so we can draw our little chromosome down here, and we can say that in this one section, it's going to have the shell gene, and in this other section, it's going to have the pattern gene, and those are going to be 13 map units away.